Hi, I'm Whitney Espich, the CEO of the MIT Alumni Association, and I hope you enjoy this digital production created for alumni and friends like you.
Hello, and a warm welcome to you all. Thank you so much for joining us today. My name is Kelly Cervic. I'm thrilled to be with you all, serving as your host and moderator for today's MIT Alumni Forum. I am a science journalist, most recently working as a staff reporter and editor at Science Magazine. I am currently on campus here in Cambridge as one of this year's MIT Knight Science Journalism Fellows. I'm particularly interested in mental health research and the role of neuroscience in society. And so it's my pleasure to introduce our speaker today, Professor Nancy Kanwisher. Professor Kanwisher received her Bachelor of Science and PhD from MIT, working with Professor Molly Potter. After a postdoc as a MacArthur Fellow in Peace and International Security, and a second postdoc in the lab of Anne Treisman at UC Berkeley, she held faculty positions at UCLA and then Harvard, before returning to MIT in 1997. She is now serving as investigator at the McGovern Institute for Brain Research, a faculty member in the Department of Brain and Cognitive Sciences, and a member of the Center for Minds, Brains, and Machines. Professor Kanwisher uses brain imaging and other methods to discover the functional organization of the human brain as a window into the architecture of the mind. She has received the Trolland Award, the Golden Brain Award, the Heineken Prize, and a McVicker Faculty Fellow Teaching Award from MIT. And she is a member of the National Academy of Sciences and the American Academy of Arts and Sciences. So we are hoping for an interactive question and answer session after Professor Kanwisher's presentation. So don't be shy, please type your questions into the text box that you will see on the lower portion of the viewing webpage. And you can type your questions at any time throughout the broadcast, and we will be collecting them as we go along. So with that note, I welcome Professor Kanwisher.
Thanks so much, Kelly. It is a great pleasure and an honor to be here speaking to you guys. Yeah, the MIT alums, we have a lot in common. At least I love this place. I imagine you do too. And I still have to pinch myself to believe I get to be here doing what I do. So let me share my screen. So I want to start with some good news, and that is that my field of human cognitive neuroscience has made a lot of progress in the last 25 years or so. And a great way to see that is to step back to consider what we knew just a few decades ago in 1990. And back then we had a very approximate idea of the location of the brain of a few mental functions from studying people who had the great misfortune to have had damage to those parts of the brain. So if you had damage to the bottom of the back of the right hemisphere down there, you might selectively lose your ability to recognize faces. Or damage to the left hemisphere in these pink regions here might lead to problems with language. That had actually been known for 200 years. Or damage to the parietal lobes up here would lead to difficulties in moving attention around in space. And so that was worth knowing, but kind of rudimentary. And that was kind of it. And then functional MRI came along. And functional MRI, as I imagine most of you know, is a method for looking at activity in the human brain that looks at changes in blood flow. The idea is a lot like the idea of muscles. If you get up and run around, you need to send more blood supply to the muscles to enable you to do that. And similarly, if a bunch of neurons in some part of your brain start firing a lot, that's metabolically expensive and you need to send more blood flow to that region. So all the data I'm going to show you is using functional MRI to look at changes in blood flow resulting from neural activity. So to see where we are now with functional MRI, there are now dozens of regions of the cortex for which we have a pretty good idea of what that little patch of brain does. And each of these regions is present in approximately the same location in pretty much every normal person. And of course, this diagram is highly schematic. What does it mean to stick some colored ovals on a brain? So let me show you some real data. So this is a bottom view of a brain looking up from the bottom at the brain of a recent member of my lab who was a great subject, so we scanned him a lot. And what we're seeing is the bottom surface with the frontal lobes in facing each other here, and the back of the head of the right and left hemispheres out there. And so the cortical surface looks smooth here. That's because it's been mathematically inflated. In the brain, the cortex is all folded up. It's about the size and thickness of a large pizza. And to fit that pizza into your skull, you need to crumple it, and that's why it's all folded up. But to see the native structure of the cortex, we want to unfold it and see that surface. So that's what you see in gray here. And the color patches are the things that we see with functional MRI looking at the function of different regions. So for example, that little blue patch right there is the fusiform face area, because we and many others have seen that it responds much more when people look at faces, that's the blue bar here, than when they look at any other kind of visual stimuli, objects, words, scenes, anything like that. And this is again, the blood flow change in that part of the brain when people look at faces compared to these other things. And yet right next door, just a centimeter or two away, is this little green patch here, colored green on the brain, because here's what it does. It responds really strongly when you look at images of scenes or places. It could be the picture of your office or a forest or a city street or a wide open landscape in Utah or something like that. It doesn't matter what the scene is. It responds exactly 0 to faces. 0 means the same as when you stare at a tiny dot, right? We can't actually turn the brain off, but for the visual system, staring at a dot is as close to off as we can get. And so that region likes scenes. Over in the left hemisphere is a particularly interesting region right here, known as the visual word form area. So named because it responds very strongly when people look at visually presented letters and letter strings or words, that's the yellow bar here, compared to all these other different kinds of visual images. And this region is particularly interesting because it's the one part of high-level visual cortex where we know that the selectivity of that region was learned in that individual's experience rather than being wired up innately. And we know that in a bunch of different ways. So first, you don't see this selective response to words and letters until after people are taught to read, not before. So that's one clue. The second clue is that humans have only been reading for a few thousand years, and that's just not enough time for natural selection to have wired up a specialized circuit like this. And the third way we know this is that if you scan adults who read maybe 2 orthographies, say English and Arabic, but not Hebrew, that region will respond to English and Arabic orthographies, but not Hebrew, and all the other combinations. Okay, moving out onto the lateral surface of the brain, we see some more good stuff. So these little purple patches here are bits that respond more when you look at images of bodies and body parts, feet, hands, knees, torsos, whatever. That's the purple bar here. Lower response to faces, that's one body part it doesn't respond as strongly to, scenes, objects, etc. Okay, so these are all aspects of high-level vision. What about other sensory modalities? Well, over here in orange, on the kind of top of the temporal lobe, is a region that responds very strongly. In fact, it's responding very strongly in your brains right now. Even if you're already bored and not paying attention, it's going, because this region responds not to the meaning of what I'm saying, but to the sounds of speech. And so that region right there processes, say, the difference between a ba and a pa, which is, if you looked at the acoustic spectrogram, they're very, very similar, but we're finely attuned to that because that will create the difference in meaning between 2 words. And so now one might wonder, okay, here's all these special bits of brain for different aspects of perception, visual and auditory perception. Is this kind of specialized bits of brain something that's just true about perception, or do we also have specialized machinery for aspects of high-level thought? And so that's a question that's been fiercely debated in my field in various versions, something like 200 years. And that's why I was super excited when the very amazing Ed Fedorenko came as a postdoc to my lab about 10 years ago. She's now a faculty member right next door. She's amazing. And what she set out to ask was whether the language regions in the brain, shown here in red, are also very specific for language, or whether they're also engaged when you do lots of other things. And so what Ev did was first figure out how to identify those regions separately in each subject individually, just by scanning them while they read sentences that carry meaning versus strings of non-words that don't have any meaning. And that enables her to identify those regions in each subject. That's important because their exact location varies from subject to subject. So if you just look in general across people, you blur that selective response. So what she asked was, would those regions respond when people are doing other things other than language? So she made up a whole bunch of tasks. The details don't matter, but lots of tasks that people had argued might overlap with language in the brain, like holding stuff in working memory, doing arithmetic, listening to music, and various other kinds of tasks. And what she found is shown here. So this is basically Wernicke's area, a language region in the temporal lobe. Here's in black is its response when people read sentences versus non-word strings. That's how we define it. So that just shows you the selective response to language meaning. And here's the response to all those other conditions, basically a big bunch of nothing. That means that region is very selectively engaged by language. And similarly, up here in what's basically Broca's area in the left frontal lobe, this region does the same thing, a strong and selective response to language and not much of a response to any of these other tasks. And I love this result because it's not just, another selective blob on the brain, much as I do like finding those. It also answers a basic question that I'm guessing many of you in this audience have already wondered, and that is, what is the relationship between language and thought? Like, do we think in language? Could we think if we didn't have language? What is up with all of that? And these data tell us that language is not the same thing as thought in the brain. All these other kinds of cognitive thinking operations activate other regions of the brain, just not the language regions. And in fact, even stronger evidence that language and thought are separate in the brain comes from the very beautiful work of Rosemary Varley in England. She has been studying for many years a small group of people who have global aphasia. This is a very horrible disorder that happens if people have big left hemisphere strokes that knock out all of this language function. These are people who not only can't speak, but who really cannot understand language at all. And so what Rosemary has asked is, can these people think in other ways? And what she's shown is that pretty much every mental operation that you might have thought would require language doesn't. So these people can do mental arithmetic, they can solve logic problems, they can appreciate music, they can hold information in working memory, and they can think about the contents of another person's thoughts. So that's pretty cool. And that also brings me to possibly the wildest and coolest patch of brain, this little pink patch here, studied in detail by my colleague Rebecca Sachs, who showed that that region is very selectively engaged when you think about what another person is thinking. That's the pink bar here. And what's impressive about Rebecca's work is how specific that region is. So if you think about what another person looks like, or you think about their cultural practices, any of these external things you can see on the outside, that region is not engaged. In fact, it's not even engaged when you think about their bodily sensations, like this person is thirsty or hungry or in pain. That region does not turn on. It only turns on when you think about the contents of their thoughts. Pretty amazing. Okay, so lest my audience think I'm a complete nut at this point, putting crazily specific blobs on every patch of the brain, I hasten to add that not all regions of the cortex conduct very specific functions. In fact, these white patches here are sometimes called multiple demand cortex. That means they respond to multiple different kinds of demanding cognitive tasks. In other words, they're very, very general purpose. They're kind of like the opposite of all these other regions that I've talked about here. So in fact, if you look at lots of different tasks, the details don't matter, but the difficult version of the task is the solid bar, and the easy version of that task is the outline bar. And Fedorenko did that across lots of different kinds of tasks and showed that these regions respond to all these different kinds of demand. Okay, so that is my whirlwind overview of some of my favorite parts of the cortex in humans. And so I think that this picture that we now have of the brain counts as real progress. So we now know there's a set of functionally distinct regions that are present in everyone, and that includes regions that are specialized for abstract, uniquely human functions, like language and music and thinking about another person's thoughts. And I think this is cool because it's sort of an initial sketch of who we are. This is the machinery that's running in your brain. It has these pieces and probably lots more that we don't know about yet, but at least these basic components. At the same time, this is just the barest beginning. And so I think really the way to think about this diagram is as a roadmap for future research. So what I want to do in the rest of this talk is survey some of the vast landscape of unanswered questions that we're trying to chip away at in my lab right now. And so one of the first ones that comes to mind is that functional MRI is wonderful. It's given us this map, but that map just shows when those regions turn on and what they turn on selectively for or during. It doesn't tell us that you need those regions to conduct those functions. And that's a problem because science is all about causality. It's about the fact that, activity in this region is, that you can't have this kind of thought without that region, or you can't do this mental process. For example, you can't recognize faces unless you have that region. And functional MRI is simply not going to answer that question. And that means we need other kinds of data to answer that kind of question. And that's why I was pretty excited when I got a phone call a few years ago from a colleague telling me that this gentleman here was in a hospital in Japan awaiting neurosurgery. And the neurosurgeons had placed electrodes all over the bottom of his brain, as shown here, to map out specific mental functions and to try to find the focus of his seizures. So usually epilepsy responds well to drugs, but in some cases it doesn't. And then the best option, if it's very severe, is neurosurgery, basically to find the source of the seizures and remove it. And so that's what this guy was in the hospital waiting for. And the neurosurgeons had electrodes in all these locations, and they asked me, and this gentleman had very kindly agreed to allow us to record from these electrodes for the week or so that he was in the hospital waiting to have seizures while he looked at different images. So the reason that that was such a precious opportunity to me can be seen in this picture of the bottom of my brain oriented the same way. So here are my face regions in red, my scene responsive regions in green, and these purple bits respond preferentially when you look at color versus grayscale. And so you can see these electrodes in here traverse a lot of territory of particular interest to me. So we sent some images to Japan, and they recorded responses from the surface of this gentleman's brain in each of these electrodes along here. So that's what you see here. Each of these graphs shows time on the x-axis and the magnitude of the neural response on the y-axis, and the different kinds of stimuli in color. So this little site 173, which is right about there, here's a response to faces, and here's a response to everything else. And so if you were kind of underwhelmed by the bar charts I showed at the beginning of the talk, it's like, oh, there's faces, there's objects. Okay, it's a little more to faces, but it still responds to objects. I think that's because functional MRI is a relatively crude measure because it's looking at blood flow that smears over the brain. When you have direct electrical activity, you see spectacular specificity like this right here. But I started off with this point about causality. And that's the real purpose of this, of talking about this, is that the neurosurgeons decided, of course, for clinical reasons, none of this happens unless the neurosurgeons need to do it clinically, to electrically stimulate those electrodes to better understand the function of those electrodes and their epileptic role. And so what I'm going to show you next is a videotape of what happens when this guy is stimulated in those electrodes. And he's speaking Japanese, so you probably won't understand what he's saying. But here he is getting stimulated, and here's subtitles explaining what he is saying. He's simply asked to say, did anything change? Guys, just the perfect subject. He's trying so hard to explain exactly what's going on. So here you see the stimulation. He doesn't know where he's getting stimulated in the brain. He doesn't even know there's a face area. He's just looking and trying to tell us if anything changed. So that shows that that region is causally involved in face perception. Question here is it causally involved in perceiving and other things that aren't faces?
No change. 最初の1秒ぐらい、始まって1秒ぐらいがなんか、横になんか目、口って感じでちょっと一瞬見えたんですけど、何かなって思ってたら、気がついたらもうこの箱にしか見えないっていうか。またさっきの横に。
This is a kanji character on a card for shortened here. Okay, you get the idea. What this shows is that that little patch of brain not only responds selectively when he sees faces, but is causally specific for faces. That is, when you electrically stimulate it, it only affects his perceptive faces. If he's seeing a face, it changes it. If he's seeing something else, it puts a face on top of it. When he was stimulated next door in a region that responded more to color than grayscale, he reported seeing rainbows on top of whatever he was looking at. Okay, so that's at least some evidence for the specificity of the causal role of these regions. Probably already, many of you have been looking at this diagram thinking, what else? What other stuff is in there? We've been thinking that too, and we're spending a lot of time in my lab testing various things. Some recent work has argued the little patch of brain about here responds not just when you see faces or hear people speaking, but when you see two people interacting with each other. In fact, you can decode from the pattern of activity right there, whether that interaction is a positive one, a cooperative one, or a competitive one. That's something that is very early developing, that 6-month-old infants can tell the difference between whether people are cooperating with each other or competing. Another region that we've been studying a lot is shown roughly here where these purple blobs are. And this region is involved in understanding the physical world. Okay, it's not like 801, 802. But short of that, before you took any of those courses, you had to think a lot about the physics of the world. In fact, you cannot take a single action on the world without thinking about the physics of the objects you're acting on. I can't pick up my phone or walk across the room without thinking about the weight of the phone, the friction of its surface, whether the floor will support my weight, whether there's a barrier in the way that I can't get past. So these basic principles of the physics of the objects and our surroundings are essential for everything we do. And we're starting to think that there may be some sort of specialized machinery up there to help us think about that. So all of the work that I've talked about so far was done by me and people in my lab or colleagues sitting around thinking, maybe there's a special brain region for this. Let's go look and see if we can find it. And that's great. That's hypothesis and driven science. Nothing wrong with it. It's very respectable. But there's another way to do science, and that is to collect a whole lot of data. And as a friend of mine says, shake the data by the scruff of the neck and say, speak to me. That's a very informal way to describe a whole branch of math in which you take a big pile of data like this. In this case, we were looking at auditory cortex, and we had the response in a big patch of auditory cortex across 11,000 three-dimensional pixels or voxels in the brain. Each of those voxels, we had the response to 165 different everyday sounds, the kind of most common sounds you hear and recognize in little 2-second clips that were played to subjects in the scanner. And so these are our data. And then we just applied a bunch of math that basically factorizes this matrix and says, let's boil this matrix down to its essence and see its basic components. And when we do that, we find 6 components account for about 80% of the replicable variance in those data. And so some of those were things we already knew, but some cool new stuff came out of this. And so here is one component that emerged kind of blind from that analysis. The analysis has no labels, so it doesn't know where the voxels are or which sound is which. But once it's discovered a component, you can stick the labels back on and say, oh, what is this? And you can see that what it is a strong and selective response to music. And that was really cool because we had looked for this before and not seen it. And you can kind of demix neural responses better with this matrix factorization response. Would enable us to discover something new. Nonetheless, I love this math, but I'm always a little nervous when you have to do a lot of math to see a result. And so I was thrilled when a few years later, we were able to collect intracranial recordings like the Japanese gentleman I showed you from people who were in epilepsy units with electrodes on their auditory cortex, where we showed, for example, a single electrode. We saw many of these, but here's one example electrode that is responding much more to music. That's all the blue and purple bars here, and lots of different kinds of music, as you can see, than any of the other sounds. And that validates both this data-driven method and the result that we discovered. And so we have another new blob to add on the brain, a music selective patch approximately there. But we also have a new method, and I was so excited about this. I thought, well, we better try this data-driven analysis on other big data sets. And that's when my fabulous postdoc, Meenakshi Khosla, came along and mentioned to me that there was a beautiful new data set that had just been released that was collected by another lab of 8 people, each looking at 10,000 images. That is a huge data set, spectacular data. And so she applied a similar data-driven method to try to just ask, what is the basic structure in this bottom surface of the brain where we see the face, place, body, and word selectivity? What will we see when we do a data-driven analysis of those data? And so what she found is 5 components that were consistent across subjects. And here are 4 of them. You can see, here's one. I don't know if you can see those images well enough, but they are all scenes. And here is the magnitude of response of that component to each of the 1,000 images that were common across subjects, color-coded by how much, how salient the scene is in that image, rated by separate people. So that's the scene selective cortex found again. Here's a face selective component found again. It's hard to see it here, but this is a word or text selective component. There are little teeny bits of text in each of these images. And here's body selective cortex. So we're basically replicating here what we already knew. And on the one hand, you might say, well, duh. But to me, this was super exciting. First, it's a strong positive control for this method, right? So positive control is you try a new method and you see if it rediscovers stuff you know to be true. And if it does, you go, good method. So that's nice. But beyond that, I love this result because to me, it means that these components that we found before, just by thinking stuff up and looking for it, they're not just weird little things we happen to look for. They are 4 of the main kind of consistent response profiles that you see in that whole part of the brain. And I think that's pretty neat. But even cooler is that there's another one. You may have noticed all this white space in the middle of the graph. And what is that other one? Well, this was a new component we had never seen before. And here it is. It is a very, very selective response to food. And I will skip all the details and just say that when we first found this, we thought, yeah, right. It's just things that are reddish or colorful or have round, curvy things because its food is sitting on plates. We had all these alternative accounts, and over 6 months, we ruled them all out and decided that it's just true. And so we have another selective response here on the bottom surface of the brain. Okay, enough with all these crazy specializations. A very different question that probably many of you are also wondering about is, how do you wire up a brain to have this incredibly intricate structure with these very particular functions in roughly the same place in every person? Well, we wonder about that a lot too. And so there's many studies that have looked at this kind of high-level visual part of the brain where the face, place, body, word, and food regions are. And they have argued that especially the face region develops really slowly and is still developing through adolescence. And there's a really beautiful study by Marge Livingstone at Harvard, who raised baby monkeys without letting them see faces. They had plenty of visual experience from human caregivers who took good care of them, but had welding masks over their faces all the while. And they had plenty of time with their parents and siblings in the dark. So they had a reasonable rearing, they just never saw faces. And what Marge found is that those monkeys do not develop the face regions like this that normal monkeys do. And so these and other findings have led people to say that at least the face regions take many years to develop and seem to require visual experience with faces. So that's a very important conclusion. And so my first reaction was, okay, maybe, but really, let's try this again. And so one problem with the developmental data is that most of the prior studies were done on kids 4 years old and up. And that's useful, that's great, but 4 years is pretty old in terms of brain development. 4-year-olds can do a lot of stuff and have had lots of experience already. So scanning infants would be even cooler. But it is almost impossible. That's when my amazing colleague Rebecca Sachs stepped in with here her then postdoc Ben Dean and her champion star subject, her own son Arthur, who was scanned many, many times in the first year of his life, and who gave us beautiful data along with a few other kids, mostly the kids of postdocs in our department, but it's expanded since then. And that enabled Rebecca and Ben to find that the face and scene responses in 6-month-old infants have a similar spatial topography in the brain. And that's really worth knowing. But one thing that was hard to tell was exactly how selective the response was in each of those regions. And that's hard because it's just so hard to get good data from infants. You know, they're squirming and wiggling and their parents are nervous and the whole thing is really, marginal. And that's when the really amazing Heather Kosakowski came along. Heather got her PhD working with me and Rebecca Sachs just a few years ago. And Heather has an ability to scan infants that is just beyond belief. She can take a screaming, squawking train wreck of an infant when I'm watching thinking this is stupid, we should just call it off. It is not happening. And Heather will get that kid to calm down, put them in the scanner and get beautiful data. So during her PhD, she scanned 83 infants, quite an amazing feat. And she found that the face, place, and body areas are all not only present by 6 months of age, but show adult-like selectivity. So here's a response in the face area in 6-month-old infants to faces and the other conditions here. The place area, the baseline's funny, but scenes are higher than the other conditions. And the body area shows a selective response to bodies. That's the pink bar here. So they're all present very early. They probably continue to develop after that, but they're present extremely early. And so what that tells us is that no, these regions do not take many years to arise, even if they keep developing after that. Well, what about this other idea that the development of these regions requires visual experience? Well, my postdoc Ratan Murthy decided to take another whack at that question in a very different way. And that was by scanning congenitally blind subjects as they felt faces. So he made 3D printed objects, as shown here, of faces and scenes and hands and chairs. And he put participants in the scanner, blind and sighted, and put a cardboard disc on their stomach and rotated it from outside the scanner so they could just put their hand on top of each object and feel each one as it went by. And what he found, first as a control for the sighted subjects, looking at these stimuli, you see the usual selective response, higher response to faces than the other stimuli when looking at the stimuli. What about the blind subjects? Here's the blind response in the same region when feeling faces compared to the other stimuli. So I find this result pretty remarkable. I will say the magnitude of response here is lower for blind subjects feeling faces than for sighted subjects seeing them. So it's not identical. And we didn't see it in every subject, though we saw it in over half. Nonetheless, it shows that no, you do not need to see faces during development to get a face selective region in that part of the brain. Okay, so most of the mysteries of development are left unanswered, but at least we know that those regions develop very early or arise very early, and that at least you get face selectivity in congenitally blind subjects. You might also be wondering about the evolutionary origins of these regions. Me too. We don't really know, but there's some tantalizing snippets. First of all, if we consider our most recent common ancestor with macaque monkeys, that would be 30 million years ago when we branched off from macaques. And we ask how similar is the organization of visual cortex in macaques and humans. It's actually really similar. So here's the data I've been showing you in humans with a bottom view like this. And this is the face regions in purple, color preferring regions in blue, and scene preferring regions in green. Here's this is on the bottom surface in humans, and here's on the lateral surface in monkeys, showing you the same order: faces, color, places. So it's way on the side in monkeys and on the bottom in humans, but it's the same order. And it's hard to imagine anyone, designing a brain thinking, oh yeah, I'll stick the color right between faces and scenes. That's where it belongs. Instead, this seems like the kind of arbitrary thing that happens from inheritance from a common ancestor. And in contrast, interestingly, I think auditory cortex may be a very different beast in humans and in monkeys. We use hearing very differently than monkeys. Some monkeys have a few words, like 2 or 3, not like us. And monkeys don't listen to music, anything like us. And so they use their auditory systems very differently. And there's an emerging picture that the organization of auditory cortex in monkeys may be quite different than in humans, unlike visual cortex, which is so similar. Okay, so the last question I want to consider is maybe the most fundamental, and that is, why should a brain be designed like this in the first place? Why, from a computational point of view, does it make sense to have specialized machinery for face recognition? Why not just have generically powerful visual processing machinery that can handle whatever comes along without special face machinery? And so for years, I would end every talk saying, the biggest question here is why, and I have no way to address it. And so I leave you with that unanswered mystery. And we still haven't totally answered it, but we now have a new way to go at that question that I'm very excited about. And that new way has been made possible by the astonishing recent success of artificial neural networks. So I'm sure this audience knows that artificial neural networks have transformed artificial intelligence. They are the reason now that your phone recognizes faces of your friends really well, even when you don't tell it to, that you can give it dictation with voice memos and it does really well. And they can do all these things. These are all artificial neural networks that have suddenly, in just the last decade or so, become extremely powerful in solving pattern recognition problems in vision and perception. And so what does this have to do with the brain? Good question. I think we can use these artificial neural networks to ask, why does the brain have special machinery for faces? And this is work with my former postdoc, Katharina Dobbs, who now has her own lab at the University of Gießen in Germany. And Katerina decided to use artificial neural networks to test a hypothesis that had kind of been around in the air for a long time, but it was just kind of talk. We didn't have a way to really make it precise. And the general idea was, why should the brain have a special region for processing faces? Well, because the features you use for discriminating faces are just different from the features you use for discriminating, you know, an apple from a dog from a toaster, right? And so if the features are different, maybe it makes sense to have different patches of brain do that. And so that's a perfectly reasonable idea. But as I said, it was, you know, just talk. We didn't have a way to test it. So Katerina said, okay, we can test this using artificial neural networks, in this case, convolutional neural networks, a subclass of artificial neural networks. And so these networks, as you probably know, are you know, basically programs in a computer that have a series of layers of processing. So you stick an image in one end and it gets processed through these many layers. And there's an output end at the top that, that has a bunch of categories, and you can train it different ways. So a face CNN, we trained the face CNN on this massive database of 1 million different images of 1,000 images each of 1,000 different identities. And when it's trained, you feed images in and it learns to say that's, you know, Joe Schmo out here. In the object network, we started with the same architecture, the same untrained network, but in this case, we trained it on object classification. So we gave it images from the ImageNet database that actually was started by Fei-Fei Li, who's giving a talk today at MIT at 5. She realized way back in the, when was that? I don't know, 2000s, I think, that what the field of vision needed was a really, really big database of labeled images. So she started collecting the ImageNet database way back then. And now you can use it to train neural networks. That's part of why these things are suddenly so effective, is the existence of labeled databases. So you can train it to recognize objects. And so Katerina made these two networks in the usual way, trained one on faces and one on objects. And then she tested them on new faces and new objects. And here's what you find. The face trained network does really great at discriminating new faces, and it does badly at discriminating objects. The object trained network does great at discriminating new objects and does badly at faces. So any deep learning person will say, well, duh, out of domain generalization, we know that always works badly with artificial neural networks. But to us, this was just a nice way to show that, yes, in fact, when you optimize a network to discriminate faces, you end up with a different feature space than when you optimize it to discriminate objects, and so you see that in these data. But then we thought there's a stronger test of this hypothesis about why face and object recognition are segregated in the brain. And that stronger test is that maybe one network should not be able to do both, okay? And so we tested that. We took one network, same architecture as this, but now we just concatenated all the 1,000 different face categories at the top and the, I forget how many, 500 different object categories at the top, and trained this network on all the different faces and all the objects that have been trained separately up here. And so they're all interleaved in training, and the network was never told, these are the faces and these are the objects. It just was fed all of these different patterns, learned the labels for each of these patterns. And so my prediction was it would do badly. And I was totally wrong. The dual trained network did just great at both face discrimination and object discrimination. It did as well as the singly trained networks did. And at first I was really bummed out because so much for my hypothesis. I really liked that hypothesis, and apparently it was wrong, and that was a drag. And then I thought, wait a minute, how is this dual trained network solving both problems? Let's look inside and see. And so what Katerina did was basically lesion experiments, like have been going on in animal neuroscience for, you know, over 100 years, knocking out different units in these networks, in these trained networks, to try to find out if different subsets of units in the networks were causally involved in face recognition and object recognition. And when she did that, she found that indeed, especially the later layers of the network, there were largely non-overlapping units that were causally important for face recognition and for object recognition. And so this network has spontaneously segregated itself into two separate systems without us even telling us that, just like we see in the brain. And I just love that because I think it's, it's telling us, okay, why does the human brain have these separate systems for face recognition? Well, probably because our brains have been optimized through evolution and learning to be really good at face recognition and object recognition. And a neural network optimized to solve both of those problems does that best by segregating them. And that's probably why we do that. So that was pretty cool. And of course, once we had this result, I thought, okay, what about other categories? We're going to predict the whole organization of the whole brain once we figure out how. Yeah, right. But we decided to start small. And this was about a year and a half ago, before we had found the food region. And so Katerina and I predicted, oh, it better not segregate food from objects, because we know that's not segregated in the brain. And so she did a dual train network with food and objects. And we also found segregation for food. And so here's the degree of segregation as a function of layer in the network. And you see for both faces and food, that segregation increases as you get to later layers. And by the way, that's cool because that's what you also see in the human brain. The early stages of visual processing in the human brain don't have special machinery for faces. It's only as you get to later stages that you see that. But when we got this result, we actually wrote in the paper, well, this is a cool method and it gives a partial explanation for why we have segregation for faces, but it makes some wrong predictions. It thinks the brain should have food specialization. And then we published that. And then a few months later, we found, oh, the brain does have specialization for food. So that's pretty fun. So this leaves us wondering how much of the functional organization of the brain we're going to be able to predict from artificial neural networks optimized for tasks, which is another way of saying how much of brain organization just reflects the tasks humans have to solve, and that we've been optimized through evolution and learning to segregate in this way. Well, we don't yet know. We're trying other categories and we'll see how far it's going. And I will just say in closing that this is just one example of one of the reasons I've actually become very excited about artificial neural networks and their ability to help us understand brains, is that I think that they're enabling us to answer not just what we see in brains and sometimes even how things work in brains, but why brains are the way they are. And I think that's super exciting. And I will just close by putting my summary slide up here and saying thank you for your attention, and I am happy to take questions.
Thank you so much, Professor Kanwisher. That was a fascinating look at some of the work that you and your colleagues have been doing. I love seeing that man getting the stimulation and trying to describe the way his perception is changing. It's such an amazing sort of validation.
It is incredible, isn't it? You know,
Yeah.
I often lament that the methods we have available in human neuroscience are so much weaker than the methods that people have to study neuroscience in animals, where we have all these different tools to manipulate just this population of neurons and so forth. But that's one case where it's so much more powerful to have it be a person telling us exactly what he's experiencing.
Yeah, very special group of people who can contribute.
Yeah. Yeah, I have friends who've tried to do experiments like that with monkeys, but you have to train the monkey to tell you one thing, like, are these faces the same or different?
Right.
And then you stimulate the face patches in the monkey, and maybe the monkey's a little bit worse at the task, but he can't tell you what they look like.
Right, exactly. Yeah. Well, I know we have a ton of questions. Now is your opportunity, viewers, if you want to let us know what questions you have for Professor Kanwisher. We welcome you to type those in the text box at the lower portion of your web screen. Just scroll down. If you have your screen on full screen mode, you might need to minimize it in order to see that text box. If you cannot think of a question to ask, that is okay. We would welcome you to still participate by upvoting questions from others that are most interesting to you to increase the likelihood that we can prioritize them for Professor Kanwisher. So I'm just going to jump in because I know that we have several already. One person asks, do areas that respond to face, scenes, etc., respond to representations, painting, sculpture, photographs, the same way they respond to the real thing? Is there a cutover between an abstracted landscape and a, quote, work of art?
Yeah, it's a great question. And we haven't explored it in great detail, but I can tell you that very schematic pictures of faces and bodies, like a smiley face, really minimalist smiley face, or activates the face area almost as much as a photograph of a face. And a stick figure that you can kind of see as a schematic body activates the body selective region nearly as much as a photograph of a body. And I think that's super interesting. We haven't tried, Van Gogh and the full space. Actually, I think there are papers on this. I mean, I would bet a vast sum. Basically, of all the data I've seen, if you look at an image and you see a face in it, doesn't matter if it's a green pepper or clouds, if you see a face in it, that region is going to be active. And so one of the things that's interesting is that I didn't talk in detail about this, but we're using neural network models not only in the way I described to ask why questions about the brain, but as models of what each of those patches of brain is doing. And those models are extremely accurate at predicting the magnitude of response of those regions to new held out natural images like photographs. But they make wrong predictions when it comes to schematic things like stick figures. And we think that's an interesting case where something different is going on in humans that is not well captured by those artificial neural networks, at least yet.
Yeah. Kind of a related question on kind of what isn't localized in the brain. Someone asks, are emotions also localized in the brain? Is that something that you've looked at?
Yeah, there's lots of work on that, and I'm kind of an ignoramus on the emotion literature, but there's a lot of evidence there's definitely particular brain regions that are particularly involved in emotions. One is the insula, which is kind of in the, I don't know, it's in a fold underneath the frontal lobe near some languagey regions, and it seems to be involved in disgust and some other emotions, but a lot of those regions are down in subcortical regions. So I'm sort of I tend to obsess about the cortex, but there's a lot of good stuff that goes down in the basement of the brain, and a lot of emotion regions down there. A region called the amygdala, which you may have heard is implicated in fear and lots of other emotions as well. And a region called, there's a whole reward system where you get neural responses when people are experiencing or predicting reward, and that can be monetary reward. Or my friend Rebecca Sachs did a beautiful study published a year or two ago where she showed that if you isolate people socially for just 8 hours, you just put them in a room and say, no, you can't use social media, you can't use your phone, you just have to read a book or something. And then you put them in the scanner and show them pictures of faces, the reward regions go. The same regions that go when people look at pictures of food after being deprived of food for 8 hours. So there's a very general purpose bunch of reward regions as well, and other fancy machinery for processing different emotions.
It's interesting to think about individual differences when it comes to those, whether we could explain kind of our emotional dispositions in terms of our response.
Yes, not to mention psychiatric illnesses, which is
I wanted to make sure.
It's a huge area of research, but I'm sorry I mentioned it because I'm so ignorant about that whole literature. But just to say, it's a big area with lots and lots of people trying to understand how various, like how addictions are clearly involving the reward circuitry. There's lots and lots of work on that, and lots of other stuff with, anxiety disorders and PTSD, looking at the amygdala and so forth.
Yeah, we had one person ask about whether you or anyone has done research with people with intellectual disabilities or autism. Are those areas that research can bring to bear?
So I did a bunch of work on autism for a while, and it is just so hard to study that I give myself kind of a B- on making any, I really didn't make much progress despite considerable effort with a bunch of people on my team who were, great people doing good stuff. It's hard for so many reasons. The first reason is that the people who are really suffering from autism, who have severe cases that really make it difficult for them to socially interact with others, those people are very hard to scan. Because scanning requires going into this, dark tube and staying still for a long period of time. And if people are not verbal or otherwise, have anxieties, it's hard to scan those people. So most of the literature on autism scans high functioning people with autism, which let's face it, is like, a large percent of the MIT community. These are the people we say anyway. No, I'm sort of kidding, but people were totally able to deal with getting scanned. And those people, it's really hard to see a difference. And so we looked and looked and looked. Are their face areas different? No. Are these kind of higher level, that like those social interaction regions? We didn't exactly look at those, but there's other higher level social processing regions. Can't see any difference. The most astonishing to me was Rebecca Sachs looked in those regions that that respond specifically when you think about what other people are thinking. Like if you had to have a hypothesis about what's going on with autism, that would be hypothesis number one. And in high functioning people with autism, she saw no difference whatsoever in the location or size or selectivity of that region. So of course, none of this means that there's nothing going on. It's in part a statement about how impoverished our tools are for seeing those things.
And maybe speaks to the complexity of that condition also, that,
Yeah, but the same is true with schizophrenia. And like basically you can't diagnose any psychiatric disorder from any kind of MRI scan.
Mm-hmm.
And so these tools, you can see some things going on, but not enough to diagnose. And so these tools are great for basic research, but it's a challenge to use them to understand clinical disorders.
Yeah. I'm going to throw a kind of lighthearted, big picture question at you. Someone asks, there's the common perception that we generally use a small percentage of our brain's potential. What do you think?
Yeah. Well, I mean, I think there is a mystery about, and this really comes from behavioral literature more than neuroscience literature, that attention is very capacity limited, right? So we really, when we think we're, texting and doing something else, we're really not. We're going back and forth fast and doing badly on one of those things, right?
Right.
And so lots of literature shows that. And so we have very severe processing bottlenecks in the human brain. And so I think that's really fascinating, because when I look at all these parallel structures, it's like, why the hell can't they all work at once? What is the big deal? Especially if you have tasks that draw on different ones, why can't you do those all at once?
Mm-hmm.
And basically most of the time you can't. And so I think that's actually one of the big unsolved open mysteries about the human brain, is why we have information processing bottlenecks, and why awareness, for example, is very bandwidth limited, right? You can't be aware of lots of stuff at once. It's, the actual contents of awareness, as lots of behavioral research has shown, is really quite sparse. And I'm actually hopeful that people working with artificial neural networks will get some insights about what it is that processing bottlenecks do for us and why we have them. But that's going to take a little while.
Yeah, absolutely. Another question that we have coming in. Why are these specialized functions in the brain so localized? One might think that non-localization might be more robust to injury, disease, or any other intrusion. Also, is more localization related to superior performance?
Yeah, really good questions. I have not seen any evidence that more localization is related to superior performance. It's very, very hard to see any difference in, say, the face area. People vary a lot in their face recognition abilities behaviorally. So the behavioral work on this is really fascinating. So if you take 100 people, randomly selected people, the 2 people who are best at face recognition are so good that they probably hide it. So there's studies of these, they're called super recognizers, and it creeps other people out. Like if you say to somebody, oh yeah, I saw you 5 years ago, you were standing behind me in line waiting for this, movie theater. People's like, whoa, they're not into that. That is creepy. But there are people who are that good, and just normal people. And the bottom 2% are so bad that they routinely fail to recognize family members. So the breadth of human face recognition abilities is huge. And the first nice thing to say is that it's not correlated with IQ. So if you're like me, that you're somewhere down on the bottom end of that ability, don't worry. Yeah. So, and to me, that makes perfect sense. It's a separate little machine in the brain. Why should it be related to your other abilities? It's not, it's a separate thing, right?
Mm-hmm.
It's also heritable. And so the behavioral abilities, so if 2 identical twins are more similar in their behavioral face recognition abilities than our 2 fraternal twins. But I've taken myself right off the track. What did you ask me? Oh, what is the advantage of localizing it? I don't know, but my guess is that it's really related to how you build the structure. And why do those things land in the same place in everybody? Like what, how do you build that? And so this sort of just pushes it back one, but I think the current best guess is that much of the long-range connectivity of the brain is present at birth. The connections between, say, the face area and remote distant regions that face information gets sent to, that those long-range connections are present early on, and they probably instruct the function of each of those regions and play a major role in why they're wired up.
Mm.
And that's probably also why generally, if you have damage to those regions in adulthood, they can't just move over to nearby cortex for the most part. I mean, it's fuzzy and there's quite a big variability in outcomes, but in general, if those regions are disrupted in adulthood, the recovery is modest.
Yeah, okay. I'll give you one more, and this one was quite upvoted. Someone was asking about how memory was stored. Is memory something that has sort of overlapped with your work? This person asks, is it chemical
Yeah, it's a great
strings or something?
But I didn't hear, or what, or something?
Well, they ask, is it chemical or protein strings or something else?
Well, so the first thing, I mean, it's not what I work on, so I don't have a lot to say, but it's a great question. The first thing to realize is that long-term memories are not just inactivity. Like you might think, oh, you get a circuit going and like it's going in some loop, and that is the memory. For some kind of short-term memories, that's a possible account, but reflect on the fact that when you go under general anesthesia, the neural activity in your cortex gets shut way, way, way down. And when you come up, you haven't lost your memories. That tells us right off the bat that a lot of it has to be structural or chemical or something.
Mm-hmm.
Right. And the standard view these days is that those longer term memories take the form of just changes in the strength of connections between neurons. But to me, the interesting questions are like, I can close my eyes and imagine my mom's face, right? Boom, there it is. I'm seeing it right now. Where did that come from in my brain? I know that when I do that, I turn on my face area because I've done that experiment and everybody does that. If you can form a vivid mental image from memory, you turn on that region. But that doesn't mean it was stored there, and it probably wasn't stored there. And where the hell was it stored?
Right.
There's some nice work from a colleague of mine, Benrik Freiwald, who studies the face system in monkeys. And he's found a system closer to the hippocampus, classic memory region, in entorhinal cortex, where he thinks those memories are stored in monkeys. And it's probably really similar in humans, but it's a hard question to answer in humans.
Yeah, thank you for giving us a 1-minute attempt. That was really, really helpful. And 5 o'clock has come upon us very quickly. So I just want to thank you so much, Professor Kanwisher, for giving us kind of this whirlwind tour of the work in your field. And this kind of brings us to the close of our program. Not surprisingly, we've had some excellent questions from this very engaged community. So on behalf of the MIT Alumni Association, Professor Kanwisher, and myself, thank you so much to everyone for taking the time out of your busy schedules to join us. And we hope you enjoyed this MIT Alumni Forum. Thank you again. Have a great rest of your day.
Fun. Thank you.
Bye.
Hi, I'm Whitney Espich, the CEO of the MIT Alumni Association, and I hope you enjoyed this digital production created for alumni and friends like you.